Folks, welcome back. I hope that you've all had a good break, and we're just going to finish up the afternoon, or this part of the afternoon, with a few more talks. Our next speaker is going to be Caeley Thacker. Caeley's from Vancouver Island. She attended vet school at Massey University in New Zealand and became involved with wild sheep and goats through the BC Wildlife Health Program with Helen Schwancha, and is doing or completed her Masters of Science project at the University of Calgary with Doug Whiteside. Caeley, welcome. Let's see if you can manage the technology.
Yeah, thank you, Susan.
Perfect, you're up.
Okay. Well, thank you for the introduction, Susan, and good afternoon to everybody. I'm going to present a summary of my master's thesis work, A Health Surveillance of Thinhorn Sheep in British Columbia and Alaska. So first I'll introduce a current definition of health in wildlife species and provide a working definition in the context of this project. I'll talk about the background of this project and why and how we conducted this research, and I'll tell you what we found and what I think this means for the Thinhorn populations we examined. So wildlife health is influenced by a combination of factors related to disease ecology, climate, and anthropogenic disturbances. As Perry mentioned, health status should be considered as the cumulative effect of multiple stressors. Historically, the definition of health contained only the absence of disease, but current thinking expands this definition to include the resiliency and adaptability of a population to its environment and the ability to sustain itself, as outlined by Dr. Craig Stephen. Little is known about the health status of thinhorn sheep, including the diseases that are present within their populations and across their range, as well as how herds are doing in terms of other indicators of health. Threats to the health of bighorn sheep populations has been identified as the most significant challenge for conservation, and while far less studied, should also be assessed for thinhorn sheep. Thinhorn sheep were presumed to be unexposed and immunologically naive to many pathogens normally carried by livestock and other wildlife species due to their remote habitat. Therefore, the disease risk to individuals, herds, and populations is considered high should contact and disease transmission occur. So we aimed to identify areas of concern regarding thinhorn sheep population health and establish a baseline to support ongoing health monitoring. Health sampling of thinhorn sheep in BC was initiated in 2016 to address concerns outlined in the Thinhorn Sheep Conservation Challenges and Management Strategies for the 21st Century, a document produced by the Wild Sheep Working Group of the Western Association of Fish and Wildlife Agencies, and to start to examine aspects of One Health where the health of humans, animals, and the environment is linked. So our objectives were to determine the exposure and infection status of thinhorn sheep to Mycoplasma pneumoniae and other respiratory pathogens, with a focus on diseases known to affect wild sheep and pathogens potentially transmitted from domestic livestock. We also aim to determine the status of other health parameters that may indicate resilience of thinhorn sheep herds. The overall health status of thinhorn sheep is a function of disease status and non-infectious indicators of health. So working with thinhorn sheep or working on thinhorn sheep presents some logistical challenges. Collection of health data in remote habitats and rugged terrains. We're often facing adverse weather conditions. Herds are often small and dispersed. And the challenge of maintaining diagnostic sample quality in these conditions has previously prevented the application of tests used for other wild sheep. We use two different methods for sample collection. The first was capture and sampling of live free-ranging sheep in their winter habitats in BC and Alaska. Sample collection from live sheep was opportunistic. We piggybacked onto the concurrent collaring projects, capitalizing on the opportunity to sample sheep that were already being handled for other purposes. In this part of the study, we sampled sheep from 5 study areas. These areas were selected based on the project design of the concurrent projects or where a health concern had been raised. The study areas are geographically separated from each other with no known connectivity between the herds based on the rut and winter ranges that were previously documented. We followed established protocols for capture, handling, and sampling of Thinhorn sheep. Individuals were captured by net gun deployed from helicopter. They were hobbled and blindfolded for restraint during handling, which lasted for under 30 minutes, and rectal body temperature was monitored. We captured and sampled 46 stone sheep from BC and 75 Dall sheep from Alaska this way. Here's a map showing the study areas in BC and Alaska. So it's a bit faint there. The second way we acquired samples was through a program initiated through the Tahltan Guide and Outfitters Association in collaboration with the province of BC in 2016. This program was established in response to concerns from local outfitters, Indigenous communities, and wildlife managers that wildlife populations were possibly declining. This provided the opportunity to collect a standardized set of biological samples from hunter-harvested stones rams. Workshops to discuss the concerns and to train guides were held, and standardized sample collection kits were distributed to outfitters and hunters. The hunter harvest samples were obtained from these management units shown here. I was not able to determine more specific locations for most of the samples returned. So looking at what we found, in the live captured ewes, we were able to determine pregnancy status by measuring serum levels of pregnancy-specific protein B. Pregnancy rates were within what had been recorded in other studies of stable populations and did not give us cause for concern. One thing to consider with this, however, is that part of the concurrent study in the Skeena region of BC was examining habitat use and timing of lambing, so ewes that were more likely to be pregnant were selected for capture. We also assessed body condition. We categorized body condition of live captured animals based on subcutaneous fat by palpation of key bony prominences on a 5-point scale. So for live captured stone sheep, there was no significant difference in body condition score between years or study areas. For Dall sheep in Alaska, there appears to be a difference in body condition score between the Talkeetna and Chugach study areas for ewes and rams captured in 2019. However, it's not significant. From visually, we can see that Dall's sheep in the Talkeetna study area have a higher body condition than those in the Chugach study area, though. Condition of hunter-harvested rams was measured and categorized in 3 different ways. Body condition was recorded on a similar scale to live captured sheep based on subcutaneous and internal fat stores. Back fat was measured at the level of the last rib, and metatarsal fat content was analyzed following the KARMA protocol developed for caribou. Marrow was extracted from the metatarsus and dried to calculate percent fat. A decreasing trend in body condition score was observed between years for hunter-harvested rams. And the relative proportion of rams scored as very fat was highest in 2016, the first year of sampling. We measured cortisol levels in feces to determine relative stress levels because it's the dominant glucocorticoid in this species. We found an increasing trend in FGM levels for hunter-harvested stones rams, with a significant difference between years. Fecal glucocorticoid metabolites were measured in Dall sheep in 2019 only, and the Dall sheep had a higher mean FGM concentration than stone sheep sampled in the same year. We also looked at hair cortisol concentration. It was measured in a sample of guard hairs collected from a standardized location along the dorsal midline at the level of the shoulders. For hunter-harvested samples, HCC did not vary by year. For live captured stone samples, HCC did vary by year. And HCC was not compared between the 2 sample collection methods, the live capture and hunter harvest, as season is shown to affect HCC. The live captured ewes were sampled in the late winter spring, and the rams were sampled, the hunter harvest rams were sampled from the late summer and fall. So stress is a non-specific response to changes in internal and external stimuli. We examined the trends in FGM relative to other indicators of health and found that for live captured stone sheep, FGM increases with decreasing body condition score when we take the year of capture into account. This pattern was not apparent in Dall sheep in Alaska or for hunter harvested samples. An almost 10-fold higher mean FGM level in hunter harvested stone rams relative to live captured stone ewes was found. The cortisol peak in the late summer and early fall may be associated with the rut, where increased agonistic interactions with other rams and rapid changes in body condition may elevate circulating cortisol levels. Internal parasite burden was determined using fecal float, fecal sedimentation. Helminth eggs were identified to the genus, and protostrongyloid nematode dorsal spined larvae, DSLs, were identified to species by PCR and DNA sequencing. Genetic identification of lungworm species was performed in 2017 only. And I should note the method of analysis varied between years for other live captured sheep, which may account for some of the differences we observed. So I did not actually compare between years for this reason. 32 live captured and 19 hunter harvested stone sheep samples were analyzed. And we found gastrointestinal nematode eggs in a higher proportion of live captured stones used than from the hunter harvested rams. In the Williston study area, or most southern study area, all 4 sheep that we captured at the lower elevation site had winter tick and evidence of irritation, including barbering along the dorsum and ventrum and lateral abdomen. Ticks were collected for identification. The 4 sheep captured at the higher elevation area at the same site showed no evidence of external parasitism. This had been previously documented in an earlier study in BC. We found no evidence of external parasites on any other live captured stones or Dall sheep. We used serum and plasma from live captured sheep and blood from hunter harvested sheep dried on Nobuto strips to test for antibodies to specific viral and bacterial pathogens. Serum antibodies to ovine herpes virus 2 were found in 89.5% of stone sheep tested. But overall, Thinhorn sheep in BC showed minimal exposure to most viral and bacterial pathogens carried by domestic small ruminants and other wild ungulate species. Antibodies to the paramyxovirus BRSV were found in 2 stones used from the Dome study area. In Alaska, however, all Dall sheep were seropositive for Toxoplasma gondii in 2019. Seroprevalence decreased to 73.9% in 2020. No clinical signs were observed that we could associate with T. gondii infection, and seroprevalence for T. gondii was 0 for all live captured stone sheep. So our most significant serological finding is the very high levels of exposure to Toxoplasma gondii in South Central Alaska. T. gondii is an apicoplexin parasite that can induce neurologic disease or abortion in the intermediate host and rarely in the definitive host. Thinhorn sheep may act as an intermediate host, becoming infected through environmental contamination with oocysts shed by infected wild felids, such as Canada lynx. T. gondii antibodies in the sera of Dall sheep and other wild ungulates was previously described, but at a much lower seroprevalence than we demonstrated. Seroprevalence typically increases with age due to increasing risk of lifetime exposure. And there's several other reasons that may explain this difference from what was previously described. One is the threshold used for determining a positive result at different laboratories and the method of analysis. Lynx hair cycles are well documented and may provide an explanation for change in the prevalence of T. gondii as well. so that the infection of the intermediate host is dependent on the density of the lynx on the shared landscape. And we know that the population of lynx either peaked in the Talkeetna Mountains area in 2019 or is soon to peak. A high seroprevalence of malignant Catarrhal fever virus, MCF, was present in stone sheep in the Skeena region of BC. MCF was detected in our study using an ELISA specific for ovine herpes virus 2. The MCF carried by bighorn sheep, ovine herpes virus 3, is closely related to but genetically distinct from ovine herpes virus 2. The ELISA used detects antibodies from both types. It cross-reacts. And so genotyping of the MCF detected in Thinhorn sheep has not been conducted. But based on recent findings in bighorn sheep, it is likely either is or is closely related to the strain in bighorn sheep, the ovine herpes virus 3. There does not appear to be significant health issues associated with this finding, and it is something worth keeping an eye on in future monitoring efforts. Nasal swab samples were collected from all live captured and hunter harvested Thinhorn sheep in this study. MOBI was detected in 3 Dall sheep in Alaska from the Talkeetna study area in 2019. And no positive results were found in the Chugach area of Alaska. Compulsory inspection nasal swabs of hunter harvested stone sheep returned suspect positive results in 2019, one from the Skeena region and one from the Peace regions of BC. These rams were not associated with the hunter harvest sample kit, or we were not able to make that connection. And so no other biological samples were able to be tested in our study. When looking at the MOBI PCR and ELISA results, MOBI was not detected on nasal swab samples from the 2 serologically positive and 2 indeterminate individuals. The presence of the organism in the nasal passages and detection by PCR does not necessarily equate with infection of the animal, and infection does not always lead to an immune response. Antibodies at a detectable level are transient, and this could be due to transient infection and subsequent clearance or subclinical infection where a possible immune response has not been shown. Indeterminate PCR results are those that show some amplification of genetic material, but below a threshold level to be considered positive. So we demonstrated low seroprevalences for most viral pathogens carried by domestic livestock or other wild ungulate species. This was not totally unexpected, as the remote areas that Thinhorn sheep typically inhabit, there is minimal contact with domestic livestock. Our findings are consistent with the results from health survey of Thinhorn sheep in the Dunlevy and Schuler herds along the Williston Reservoir, which Helen previously mentioned. And the low seropositivity to many common viral pathogens of domestic species indicates that the associated diseases are not circulating in Thinhorn populations, and therefore are not having population limiting effects at this time. However, as Thinhorn sheep are immunologically naive to many diseases, if introduction does occur, there is a risk of high morbidity and potentially mortality. 26 species of bacteria were isolated from Thinhorn sheep swab samples, tonsil swab samples. Of the species previously shown to be implicated in wild sheep polymicrobial pneumonia, incidence differed by study area. B. triolosi was not isolated from any sheep in the Skeena region of BC, but was found in 62.5% of the Williston sheep and 58.7% of sheep in Alaska. Mannheimia species were isolated from 61.5% of the sheep in the Dome Mountain study area, but only 8% in the Cassiar area. The status for trace minerals list is compared to reference ranges for domestic sheep. Mean serum copper was significantly lower in both study areas than what is reported to be normal or adequate. Mean iron levels were higher than reported adequate levels, and mean zinc levels were below normal. Selenium was within normal range for the Dome study area, but deficient in the Cassiar study area. As with the live captured serum trace mineral results, copper levels were low in the Hunter harvested RAM samples. So we used liver and kidney samples. Of the 42 liver samples, 1% had copper levels considered to be deficient for domestic sheep. Mean zinc levels in kidney and liver were within normal range. However, the mean was within normal range. However, almost 20% of the samples had concentrations above the reference range. Very high cadmium levels were found in 1 kidney and 1 liver sample, that starred value there. Trace mineral levels of forage are influenced by climate, substrate type, and mineral composition and rainfall. Within a population, trace mineral levels can differ by sex due to habitat choices based on social structure. Serum trace mineral reference ranges have not been determined for free-ranging thin-horn sheep. So here we use reference intervals from other ovis species and previous findings in bighorn sheep. From this, we see potential deficiencies in BC's stone sheep in the Cassiar and Dome areas. However, our findings are similar to those from free-ranging Dall sheep in the White Mountains of Alaska for iron, selenium, and zinc, but much lower for copper. We did not detect clinical conditions that are often associated with trace mineral deficiencies. So in conclusion, our notable findings were the low seroprevalence for pathogens common to domestic ruminants. T. gondii seroprevalence in Alaska was high. MCF seroprevalence in BC was high. And there's potential trace mineral deficiencies. We found that cortisol levels were associated with body condition. This demonstrated naivety to many pathogens common to domestic livestock. And we were not able to detect one single significant health challenge with associated clinical signs. We established a point-in-time baseline for various health parameters on which to build further surveillance efforts. From this, I'd like to thank my supervisor, Dr. Doug Whiteside, for his guidance and support throughout this program, as well as my committee members and mentors, Dr. Helen Schwancha, Dr. Catherine Ruxtel, and Dr. Tom Loheis. Thank you to the many people who are involved in different aspects of this project, including the crews that I was fortunate enough to work with and who kept us all and the sheep safe. This project would not have been possible without the generous support from many organizations. Thank you.
Thank you very much, Caeley. Unfortunately, we're out of time, so we can't take any questions, but thank you for really dealing with a data poor species and adding some significant data to that. So applause.